So today we are going to study about the plasmids. Plasmids are extra autonomously replicating genetic material. Now see here, when we are saying it is extra, means it is not controlling any metabolic activities of the bacteria and it is autonomously replicating. Autonomously replicating means it will do the replication, but its replication is independent from the bacteria. Is it clear? Now, if you remember this diagram of the bacteria, three layers of the cell envelope, and this was the genetic material of the bacteria, which we are calling it as nuclear or genophore. Now, this is your plasmid, which is extra. Okay. Now, see here, they are having oricite, and oricite helps in DNA replication. This is the site from where the DNA replication will start. Now, different types of plasmids, depending upon what the gene they are having. If they contain the gene for fertility, we call it as F plasmid. So, F plasmid is a plasmid which contains the gene for fertility. And if F plasmid is there, the bacteria will work as a male or F plus. F plus or male, or it is also known as donor. So, we can say it is a non-chromosomal mechanism of sex determination. They are not chromosomes, they are only DNA, and it is a mechanism of sex determination in bacteria where if F gene is present, we call that bacteria as a male or it is donor. Another is R plasmid. R means resistance to antibiotic. So if this plasmid is present, then bacteria is having one benefit. That benefit is this, it is providing resistance to particular antibiotics. Now see, this is a plasmid which is PBR 322, means it is made by Bolivar and Rodriguez. So P stand for plasmid, okay, B stand for Bolivar, and R stand for Rodriguez. So they have observed it is having two sites. This site is resistance to ampicillin, so ampicillin resistant site, and this site is resistant to tetracycline. These are the two antibiotic resistant sites. Is it clear? And here it is having oricite. This is the site for the origin of replication. So plasmid, if they contain antibiotic resistant gene, see here once again, these antibiotic resistant gene, then they can be very important tool for our genetic engineering. Okay, so this we use them in the genetic engineering. And genetic engineering is a process by which we are transferring our desired gene and they can help in working as a selectable marker. Now see what I am trying to explain you. See here, these are the two antibiotic resistant genes. Okay, one is ampicillin, one is tetracycline. Now if suppose, see the board carefully, if suppose I put my gene in tetracycline, I've put my gene here. This is my foreign gene. Okay, so this gene I have kept it. As a result, what I will observe, this plasmid has lost tetracycline resistance. Okay, but it will be having ampicillin resistance. So now, one thing here, the plasmid which has taken my foreign gene, we will call that plasmid is recombinant plasmid. Recombinant. Recombinant means, I'm just drawing once again the diagram. I hope you will be able to understand me carefully. They have taken my foreign gene. This is the foreign gene present in them. This is your non-recombinant, which has not taken my foreign gene. Now we will observe that recombinant has lost tetracycline resistance. But non-recombinant has resistance for both. It is having resistance for tetracycline and is having resistance for ampicillin. So isn't it working as a marker and helping us to identify which has taken our foreign gene and which has not taken our foreign gene? So I hope this is clear. If the plasmid is having resistance to antibiotic, it is a very important tool for genetic engineers because they can work as a selectable marker. Okay, they are working as what? They are helping us to select, so we will say they are working as selectable marker. Detail you will be studying in your 12th class topic, biotech, but just try to understand here, because of the gene, antibiotic resistant gene, we are able to use them as a selectable marker in our genetic engineering process, a process by which we can transfer our gene. Another type of plasmid is TI plasmid. TI plasmid is a plasmid which is present in the Agrobacterium tumefaciens, and it is also a natural genetic engineer. Okay. This is the genetic material and this is the plasmid. Okay. It is double strand. You can imagine this way. Now it will actually cut this part of its own. This part will be cut, and imagine this is your plant. Okay, this is your dicot stem plant. Let us assume any plant. So this is a cell, and inside the cell this is the DNA. I'm just drawing it this way. You know very well it is not open this way only. It is having the things, nucleus and all. Now what it will do, it will cut this part and it will send it here. And as a result, this part will reach here. So it is doing by itself. No one is cutting it, no one is doing it. It will cut this part and it will send here. So we say it is a natural genetic engineer. Agrobacterium tumefaciens is natural genetic engineer. Now this plasmid, when it reach there, this part, okay, this part, focus my word carefully. This part is known as tDNA. tDNA, why? Because it is the one which is going to cause tumor. The tumor causing genes are present, that is why we say tDNA. As a result, this plasmid, okay, this complete plasmid is known as TI plasmid. TI plasmid means, I'm writing it here, you can check it. TI plasmid means tumor-inducing plasmid. So this is the plasmid which is responsible for causing the tumor, because tumor-inducing plasmid has tDNA. Is it clear? It has tDNA. I hope this line is clear to you. The Agrobacterium tumefaciens is having a plasmid which is known as TI plasmid, and TI plasmid has got this part of DNA which is known as tDNA, which is cutting by itself and is sending there and responsible for causing the tumor. So another type of plasmid is degradative plasmid, which is prepared by Anand Mohan Chakravarti. He has prepared Superbug, and the Superbug was Pseudomonas putida. So degradative plasmid is prepared by Anand Mohan Chakravarti. He has prepared Superbug, which is Pseudomonas putida, and the plasmid is present, can decompose all the hydrocarbons present in the oil spills. So we can say this plasmid is used for bioremediation. Bioremediation is a process in which plasmids or genes of the bacteria, they are used to clear the pollutant. Now another type of plasmid is called plasmid. It releases a chemical colicin to which is toxic to another bacteria. So I hope all the types of plasmids and what are plasmids, types of plasmids, Agrobacterium tumefaciens plasmid, degradative plasmid, all the topic is clear to you. Thanks for watching class. Still, if you have doubt, you can message me.
If you liked it, please give it a thumbs up and subscribe to my YouTube channel.